Welcome to Study Loop.
So in this video, we're going to be talking about epigenetics. And epigenetics is just something that's going to alter genes without affecting the nucleotide sequence of that gene. So it's able to turn genes on and off, but it's not affecting the sequence. And this can be done a couple different ways. So this can be done through methylation or adding a methyl group. It can be done through acetylation or adding an acetyl group, or also phosphorylation. And this would be to histones, which I will draw a picture of in just a second. And then also you can methylate DNA as well. So there are two kind of configurations, and we won't get into too much detail, but this is just going to explain why some genes would be on and some genes would be off. So these little circles I just drew are the histones. And then around the histones, you'll have the DNA wrapping. And in this first example, you can see that the DNA is wrapping pretty tightly. And then in this second example, you can see that the DNA is wrapping pretty loosely. So in this example, in the first one, transcription factors don't really have the room to come in. I'm drawing an X here, showing that they can't come in to bind and make proteins. While in this case, you can see that this whole section right here is exposed, so transcription factors can come in and they can bind and make proteins. So this would be considered an on configuration, and this would be considered an off. So ways or epigenetic modifications that would cause this off configuration would include DNA methylation, histone methylation, or histone deacetylation, so taking away an acetyl group. And then, so if you had a gene in this off position and you wanted to turn it back on, you would just do the opposite. So you would need DNA demethylation. You would need histone demethylation. And then you could also have histone acetylation. So that would turn these genes back on so they are active and can be transcribed and make proteins again.